disease is dictated by three factors. So you've got your pathogen, and this can include factors such as virulence, abundance of the pathogen, your host, so susceptibility or resistance, and then environment, which is conditions favorable for disease. So if your pathogen and host are in close contact, but the environment's not ideal, you're not going to see a whole lot of disease. If you're following your rotations, there's no pathogen or low levels of pathogen, or you've got a resistant variety of crop growing, then you're not going to see a whole lot of disease. And then if your pathogen and environment are there, host isn't present, then you're not going to see a lot. But if everything's present in the right amounts, you can get severe disease, right? So it's a lot of factors that all come together to result in plant disease. So why do we care? Of course, we want to have healthy plants that are able to grow throughout the growing season and result in whatever you're going to be harvesting from it. So they carry out all these functions here. Let's see, can you see my mouse here, where I'm moving the mouse? Kabir?
Hello. Now I see it. Yeah.
You see the mouse? Okay.
Yeah, I do see the mouse.
Okay, so I'll just point at things with that. In the roots, we've got nutrients and water being taken up, getting translocated throughout the plant with the vascular system. Light and carbon dioxide are transpiring in the leaves and photosynthesis, and that's all occurring in a healthy plant. But of course, pathogens interfere with each of those functions, and that results in a reduction in plant growth and possibly even plant death. And so that's what we want to avoid when we're looking at plant pathogens. Another thing that we're always talking about is life cycles of pathogens. So every plant pathogen has a different life cycle. And they're cyclical around the crop season, of course, cycle, right? And some are monocyclic, where you have one single infection event occurring. That's especially the case in Fusarium head blight, where infection occurs at anthesis. But you can have polycyclic infections as well, especially with foliar leaf pathogens. And that's what I'm showing here in this life cycle. This is Pyrenophora tritici repentis, which causes tan spot in wheat. And so, you can get that inoculum building up as the growing season goes on. One of the important parts about the life cycle is where it overwinters. So that's in this part of the life cycle here, where you get overwintering on stubble. That's often a major part of overwintering for plant pathogens. You've got infected seed as well for seed-borne pathogens, which is the case for Fusarium graminearum, and infected volunteers here as well. And so when we're following our best management practices, we're trying to stop this life cycle from occurring. So, we can follow crop rotations, control volunteers to prevent those from being a source of inoculum. You can treat infected seed, and that prevents infected seedling from occurring. Or if you're using clean seed, then you're not going to have that initial source of inoculum. As far as testing goes, there's two general methods for testing for seed-borne pathogens, and they both have their own pros and cons. First is molecular testing, so this includes conventional PCR, qPCR, ELISA, DNA sequencing, and many more. And basically, especially with Fusarium graminearum, what we're looking at is specifically the DNA for that pathogen. And the advantage to this is that it can be more sensitive, more specific. We're able to test a lot more seed, and so detect lower levels of pathogen that are present on the seed. It can be more specific. So in the case of like blackleg testing, we're able to determine what races are present, right? We can do the race analysis there. And it's also, what most growers see as an advantage is a quick turnaround for results. So normal DNA test is going to be 24 to 48 hours is what we say. And so if you need to know right away if you've got pathogen present in your seed, the DNA test is better for that, because it can give you that answer really quickly. but it does only give you that answer, yes or no, the positive or negative result, rather than a percent infected seed, which if you've got a pathogen already established in the area, that's often more important information. Traditional disease testing, which is what I do here at the lab, which is taking sterilized plant tissue, and the case with seed testing, of course, that's seed. We soak it in bleach. I'll go over the process in the next couple slides, I guess. And then we put it onto a fungal growth medium. And the advantage here is that we can test for a large number of fungi in a single test. So you can see in this image here, there's a whole bunch of stuff growing. We don't have to be targeting just a single one. With DNA testing, they're using very specific primers just for those organisms that they're looking for. And another advantage is that it does give you that percent infected seed result. So in a normal test, we do 200 seeds, and we can say two out of these 200 are infected. So that means you've got a 1% infection. And you can use that and compare different results to make a decision. On the downside, it does depend on the skill of the analyst. With few exceptions, this testing is not accredited. So, there's no organization that is out there, a third party, that's checking our testing to make sure that us and other labs are, having the same results. Contamination can be a problem. Right here in this lower right image, that's rhizopus there. This is a really common fungus. You'll see it growing on bread or other food that you leave out for too long. And it's very airborne spores. So it's in the air all around us, and once it colonizes a plate, it takes over the plate in a matter of a couple of days. So it outcompetes everything else that's on there. So we're always trying to keep contamination low. Another downside is it is a longer wait for results. So 5 to 7 days for most testing. We just have to let those fungal colonies grow until a point that we're able to identify them. And some other pathogens take even much longer than that too. Whereas, with the DNA test, you're looking at 24 to 48 hours. And some pathogens don't grow on artificial media. So we need to do that DNA test like with clubroot, right? We're not able to culture that onto a plate and determine if it's there. So in those cases, we definitely need to do DNA testing. So the process to do the traditional disease testing onto a plate, we take the seed, we mix and divide it, and try to take a representative subsample from that into a T-ball. And then we take that T-ball, we put it in 2% bleach for 5 minutes, and that's called surface sterilization, and that removes mostly bacteria from the outside of the seed, but other things that the seed may have been in contact with, so that we're getting down to that systemic internal seed-borne infection once we analyze the seed. We dry it in a laminar flow hood. This is a sterile environment. This takes up to 3 hours sometimes. And then we put it onto a fungal growth medium. We use PDA, Datodextrose agar. This is a broad spectrum fungal growth medium. Most fungi like to grow on it. It's pretty much just potato, sugars, and jello. So we do 10 seeds per plate. We do 20 plates, about 200 seeds total. So we can determine that percent infection exactly. Then we incubate it just around room temperature, about 22 degrees Celsius. We have specific lighting conditions, 75% white light, 25% UV light, and that puts the growing fungal colonies under a little bit of stress, and it induces them to produce spores, because often those spores are the diagnostic feature that allows us to differentiate different colonies. And then they grow for 5 to 7 days, like I said. And then after that 5 to 7 days, we look at the colonies under dissecting stereoscope and a compound microscope, and we're looking at the colonies, just what they look like, what the color is, and then if we need to, we'll look at the spores to determine the exact species. And with Fusarium graminearum, You know, when we're looking at a plate, this is mostly Fusarium graminearum on here, with the exception of this one colony here, which looks like a different Fusarium species. We always check the spores because there are some species that look very similar to graminearum, but the spores look quite different. So and we want to make sure that if we're reporting that it's Fusarium graminearum, that it actually is, and it's not a different species. So like I said, we can also just look at everything else that's present on the seed that grows on PDA. So we do have a test called a fungal screen in cereals. And with this, we divide it up into different categories. Of course, it does include Fusarium graminearum and other Fusarium species, which is really important because when we're looking at the seedborne aspect, you know, these are causing similar sorts of issues, seedling blights and root rots primarily. But it also includes Cochleopolis sativus, which causes common root rot, and Pyronophora and Septoria, which can be seedborne, and they are, those foliar pathogens, like I went over that life cycle of tan spot in wheat earlier. We also look at saprophytes that are present. So on these plates here, most of those kind of olive green colonies are alternaria. So these can be present at quite high amounts. It's not unusual to be upwards of 60% on a sample or even higher. And all of these, alternaria, cladosporium, epicoccum, the primary issue that we see in seed is that it causes discoloration of the seed. So it can be called black point or mildew, and it can result in downgrading if you're looking at it from a grain perspective, right? But they don't usually cause any sort of issues with if you're looking at it from an end use for seed, because they're not pathogens strictly, so they don't cause any sort of disease in a growing plant. They just break down dead and dying plant material as saprophytes. You'll see them as you're going through the field closer to harvest. These would be called sooty molds. So these are present on the heads, and they're kind of that black mold that you see growing on the entire head. But yeah, they're just kind of on the surface in those cases. With storage molds, we look at Aspergillus and Penicillium, and these can be issues if you've got moisture in your storage conditions, and they can rapidly spread, and you'll see the germ just dropping off as well in those cases. But they also produce harmful toxins. So if we detect higher amounts of these, then that's a big issue as well. But usually they're present at low amounts. So this is what Fusarium graminearum looks like. As I said, the spores are quite distinctive from other species. In these images here, you can see these darker colors on the colonies. That's the sporodokia. So that's where these asexual spores are being produced on those colonies. So that's where we make our microscope slides. Again, we want to keep in mind the life cycle of Fusarium graminearum, really important to consider when we're talking about how to manage it. So the primary infection that you're going to be seeing for Fusarium head blight is coming from these sexual spores, the parathecium, that are forming on previously infected crop residue. So under the right conditions, usually moist, a few days after maybe a rainfall event, you're going to get these spores forcefully shot out of the parathecium, and this happens at very quick. I think I did the math on it, and it's like over 100 kilometers an hour that they shoot out of there. And these will, float up in the air. They can travel some distance, and at anthesis, they'll infect, the anthers that are sticking out and go into the head, into the seed that's forming. And then once that infection has occurred, you'll see it spread from the center and out in the head. And then as that progresses, you'll see the conidia, which are the asexual spores, especially with increased rainfall occurring, they'll get splashed down lower onto the plant, and they'll go into the stems, especially at the nodes, right? So that's why there's a picture of a node here. And when we do stem testing as well, we'll often plate those nodes because that's where it's located primarily. And so that's how it's able to infect future years. But of course, it does go into the seed, and that's where we see it. The problem is that it does produce mycotoxins as well, but it can be a source of inoculum for areas that Fusarium graminearum hasn't established, or it's if you have really high amounts, it's going to be causing seedling blight and root rots, which are a problem as well. So in the field, this is what it looks like. You've got prematurely bleached heads. Often it starts kind of in the middle and spreads out from there. You'll see that orange to pink discoloration possibly, which are those asexual spores, the sporodokia that are forming that's able to, spread throughout the rest of the plant and infect the stem for future years. But there's a whole bunch of other stuff that can look kind of like Fusarium head blight. And of course, other Fusarium species that can cause Fusarium head blight. So, if you see it in the field, you can send in some plant samples, send in the heads that are showing symptoms, and we can tell you this is Fusarium graminearum, or it's Fusarium comorum, or it's something not even Fusarium, right? So, of course, it causes shriveled up kernels called FDK, Fusarium damaged kernels, or tombstone kernels, and these can have high levels of mycotoxin, which is a big issue, called deoxynivalenol. It's called vomitoxin as well. And Fusarium graminearum, other species do cause Fusarium head blight, as I said, but Fusarium graminearum causes, it's the most aggressive. It causes the most yield losses in the field. It produces the most mycotoxins. So that's why there is kind of a focus on Fusarium graminearum over those other species. So that's what the molecule of deoxynivalenol looks like. The reason that we don't want it present in feed or grain is that it does cause reduced feed intake. Of course, vomitoxin, vomit is right in the name. And it's especially a problem in non-ruminants. But it's also a big issue for malting barley as well. So they have pretty much no tolerance for DON in malting barley because it causes this sort of foaming effect here that you can see in the lower left. It does result in yield loss. So you get those small shriveled kernels, right? Extreme conditions to get 40 to 50%, but that can happen if you've got a really bad infection. The biggest loss that growers see is that downgrading that you'll have. So this was something that was published, I think, in 2018 from the government of Alberta, and they look at the cost of a Fusarium outbreak in Alberta. And the real big impact is the value of the grade loss. So going from a number 1 to a number 2, it's 7.4, or I guess the revenue loss here is $11.66 per acre. Number 1 to number 3 is 35.44 or 0.11. And then going from a number 1 to a feed, you're looking at almost $101 per acre revenue loss. But yeah, the biggest impact is this value of the grade loss to that. The yield loss is not too big in the end. And yeah, it costs a lot of money overall to the entire industry, 50 to 300 million annually since the 90s, and 4.4 billion losses in 2011 for the US. The seed-borne aspect of Fusarium graminearum, if you're looking at taking that infected seed and putting it in the ground, the biggest impact that you're going to be seeing are these sort of symptoms. So seedling blights, root rots, crown rots, that's the impact that you're going to be seeing from planting that. You're not going to be seeing necessarily Fusarium head blight if you plant infected seed, right? Because it's in that seed and, it needs that infection up into the head later on in the season for Fusarium head. So when should you do your disease testing? Right after harvest is a good time. Gives you a baseline from where you're starting, what to work towards. Is it worth it to clean? Do you have really high amounts? Is it at an amount where it's worth it to find a seed treatment that's effective for what you have? Do you need to find new seed altogether? And then I also recommend testing multiple bins or other seed options. Even seed harvested from the same field that might be in multiple bins, you can have differing results because of the distribution of disease in a field is not uniform, right? So you might have a lot of disease in one part of the field versus the other. And so you can have different amounts there. So, and if you need to compare different amounts, that's where you can make decision, use this seed lot versus this seed lot. The next time to do it is after seed cleaning. You might have a change in your level of infection. Those FDK can often be cleaned out. And the storage might change the level of infection. As time goes on, storage conditions are not favorable for fungal storage conditions. So the viability of Fusarium graminearum is going to be going down over time in storage, usually, as long as you have good storage conditions. And so you want to know what you're putting into the ground so that you can, take other management decisions as well, such as, increasing your seeding rates. I'll go over this in the best management practices part, but you want to have the most information that you have so that you can make decisions. Okay, so into the statistics part of the presentation. So when we're looking at the amount of Fusarium graminearum present each year, it's really tied closely with the amount of precipitation that we saw each year. Like I said, those spores are released, a few days after a rainfall event. So the more moisture that you have, it favors infection a lot more, and we see those peaks associated with wetter years overall. So this chart shows the percent of samples that are testing positive on the plate method. It includes all of the DNA tests as well. Most DNA tests that we get that test positive go for a follow-up plate test, so that we can determine the percent infection as well. And so this includes pretty much everything that comes in for a Fusarium test into the lab, whether it's DNA or plate. And this is throughout all crop types that we test, all cereals across all of Alberta as well. So in 2007, you can see it was minimal. 2008 started to increase. 2010, we reached kind of a peak here over 5%, and then a drop down in 2011. Continued that rise 2012, and you'll see geographically 2012 was a big change in the province. 2013, we reached a high amount. 2014, similar to 2013, then dropped down in 2015, which was a drier year. 2016 was wet year. It was really bad. That was the highest percentage of samples that tested positive in Alberta. Over 1 in 5 samples were testing positive that year. But 2017, 2018, drier years, dropped back down. And 2019, we saw another peak here. 2020, it dropped down. 2021 was another low year, quite dry. 2022, we saw an increase. And now 2023, we're seeing quite a high amount testing positive, at about 18% of samples testing positive for Fusarium graminearum. The situation this year is quite different geographically, which you'll see on the map. So this is only the third highest amount, though, after 2016 and 2019. So this year is definitely a year that Fusarium graminearum is causing some issues. When we look at the crop type, generally it breaks down by the susceptibilities of the different crops. So this is the percent of samples that are testing positive for each one. Durum is generally more susceptible by quite a large amount. to Fusarium graminearum. You can see in some of these years it was over 60% of samples that were testing positive. This year, because of the geographical distribution and where durum is grown, it's actually quite low. So only about 15% of durum samples are testing positive this year. Next up is the wheat in the blue here. That's the next most susceptible generally. And then barley is usually less susceptible in the red, but you can see this year we've had a little bit of a different situation where more barley samples are testing positive than wheat, wheat is at about 20% and barley is about 24%, it looks like there. Oat is the most resistant to Fusarium graminearum, but this year you can see we've already had, quite a few samples testing positive, a higher percentage than most years. Looking at the total average percent of infection, so this is of all of the seed that we test in the total year, thousands, thousands and thousands. What percentage of those seeds have a Fusarium graminearum infection? So it looks a little bit different, this one. The peaks were reached here in 2013, interestingly, but 2016 as well. 2016 had high amounts of infection as well as a high percentage testing positive. So that's where we see those real peaks. This year we are seeing it increase. We're around similar levels to 2019. Looking at just positive samples, what is the average percent infection in the positives year to year? So this is out of those 200 seeds that we test, what percentage of those in an average positive sample have Fusarium graminearum? So this year we're at about 2% in the average sample. This has really been driven by a specific crop, which you'll see on the next slide. But it is up from last year, similar to kind of 2021 levels, which, although it wasn't as high percentage of samples testing positive in 2021, where it was a similar amount there. So this year we're seeing that big increase in barley. So not only are we seeing more barley samples infected, it's often quite severe. So we've seen a few samples that have been up around that 30% range, and that's really skewing these results, but it's also significant, certainly. Whenever we see a sample come in that is quite high percent infection, chances are that it's barley this year. Again, durum, much lower than it usually is. Wheat is similar to last year, I would say. But yeah, it's interesting because barley often, for many years in the red here, it has been higher than wheat. So whether or not it's how it spreads within the individual heads or throughout the field, seems like if there's a barley infection, chances are good that it's going to be a higher percent infection than in wheat. For the geographical distribution, so this is the percent of samples that are testing positive, that we can identify as coming from each county, based on the customer information, that are testing positive for Fusarium graminearum. The darker the red color, the higher percentage are testing positive. So in 2007 here, it was quite isolated to southern Alberta. We had a little bit up in Barrhead testing positive even back then, but it's pretty much just in southern Alberta at this point. 2008, we see that increase, see a little bit in Rocky View. 2009, see it spread a little bit more, lower levels, so overall nothing as high. Here we are with some positives showing up in Mountain View and Acadia. Very high amounts showing up positive in Tabor at this point. Here we are in 2011 with lower levels throughout southern Alberta, but still isolated to southern Alberta. 2012, this was the year that it really spread throughout much of the province. So we're seeing a lot more areas that Fusarium graminearum is showing up in, and we're seeing really high amounts in southern Alberta here. Again, Tabor's kind of the focus there. Here we are, 2012, or 2013 continues to increase in southern Alberta. Now we're starting to see some counties in, around the Edmonton area that are at elevated levels. 2014 here, now we're starting to see in the east of Edmonton here along the Saskatchewan border that those levels starting to increase, and kind of a gradient as we move west. Again, southern Alberta, really high amounts coming out of there. Here, the levels went down overall, and southern Alberta still is the highest percentage testing positive, though. And then 2016, this was the worst year for Fusarium graminearum throughout the province. So we're having really high amounts coming out of southern Alberta, quite high amounts along that eastern border, and then you can see that gradient as you go west again. But we're also having positives showing up in Smoky River pretty consistently this year, and up in the north here in Mackenzie there. So 2017 here, levels went down. Interestingly, we're starting to see, the highest amounts or the highest percentage positives are coming from Barrhead and Westlock. We're still getting Smoky River positives here. Here, the levels kind of decrease throughout the province. Then here we're seeing along that eastern border, that's the highlight now in this year, where the southern Alberta is lower levels at this point. So things totally shifted in 2021, I think this is. What's going on here? Okay, so this is 2023 here. And so now in 2023, we're seeing a lot of positives coming up, to the west of Edmonton and the northwest of Edmonton. So Barrhead, Westlock, Parkland, but even going east as well. This is, kind of surrounding the Edmonton area is really where the highest percentage of samples are testing positive. Flagstaff, down where you guys are located, that's also an area that we're seeing more positives showing up. We've already had some positives out of Smoky River as well. But southern Alberta, because of the conditions this year, was quite low percentage that are testing positive. And as I said, the conditions through the growing season, that's really the reason that we're seeing this change in the geographical distribution. So most of the prairies, it was a pretty dry season that you can see here. This is the average precipitation in the 60 days previous to July 31st rather. So you can see most of the prairies, it was quite dry. But around the Edmonton area, and especially going to the west and the north, it was wetter than average. So that's a big reason that we're seeing higher amounts in those areas. And you can look at, we have Alberta Fusarium risk map that you can find at Alberta Agriculture. And this is really useful. And hopefully this works okay. This is kind of the risk over the month of July in Alberta. And I captured it every day at about 2 p.m. And you can see how it changes throughout the month. So it's, a lot of it is pretty low, but we are seeing, to the east of Red Deer here at this point. At this point, it really changes quite a bit, where we're seeing to the northeast of Edmonton and going down to the southeast, much higher risk. And then here, late July, we see it to the northwest. So that's why we saw those areas with a lot more Fusarium graminearum showing up. How does Alberta compare to other provinces? We're still Still a lower percentage of samples testing positive. You can see Alberta in the blue along the bottom here, Manitoba in the green, and Saskatchewan in the red. 2022 was a peak for both Saskatchewan and Manitoba, and it's gone down since that year. Saskatchewan is about 30% of samples testing positive. Manitoba is about 35. So 1 in 3 samples out of Manitoba are testing positive for Fusarium graminearum, which is much lower than the peak here that we had for 3 years between 2015 and 2017, where we're having over 90% of samples testing positive. 2021 was a year that we saw a big decrease, actually, and it was pretty similar to Alberta in both of those provinces. So good news for Alberta and Saskatchewan. I mean, for Manitoba and Saskatchewan, makes sense based on the drier conditions in those provinces. And the average infection of positives in those areas is actually lower this year than in Alberta. So, I showed this before. Alberta is about 2% in the average sample that is positive. Manitoba is just under that, and Saskatchewan is even less than 1% in the average positive sample. So again, when we see a sample come in that has a high level of infection this year, it's definitely more often been from Alberta compared to some other previous years where 2016 we had this huge amount in Manitoba where the average positive sample was over 18%. So it's good news for them, as I said. So what does it all mean? What are the thresholds? Unfortunately, there's no clear established thresholds. It's not an accredited test. The one exception is true loose mud and barley, where we have the CFI grading tables because it is an accredited test, right? So on the table 2 here, you can see the maximum percentage of true loose mud that's allowed in barley, and it's between 2 and 4%. It's 2% for number 1 for pedigreed seed and 4% for number 2. Or 4% for common seed and 6% for number 2 common seed. But with Fusarium graminearum, I mean, look at the sample. Is it acceptable? This sample is actually 25% infection. So about 1 in 4 seeds has an infection. So that's why it's really important to do a seed test, is because if you're looking at this, it might look okay. There's a few shriveled seeds that I see in there, but the actual number of seeds is quite high. And in this case, you wouldn't want to use that seed most likely. Based on the provinces, Manitoba's recommendations, it is important not to use seed with high levels of Fusarium. They don't really say what high levels are. So those recommendations are a little bit difficult to make decisions based on. In Saskatchewan, it's a lot clearer what their recommendations are. They have this little flow chart that you can find on the Saskatchewan's website. And basically it comes down to whether or not Fusarium graminearum established in the area. If it's less, or if it's more than 5%, they say don't use the seed. But if it's total Fusarium is over or less than 5%, and F graminearum's less than 5%, then use a seed treatment, is what they say. But if total Fusarium is less than 10%, they don't even recommend using a seed treatment for that. They do stress that it comes down to a large number of factors, and they say it's a rule of thumb. So it's not, you don't need to follow these recommendations necessarily. It does come down to individuals and the risk that they're willing to take. And that's kind of where my recommendations come into, what I recommend. In Alberta, it was very clear from 1999 to 2020, we had 0 tolerance for Fusarium graminearum in seed. You can sell or plant any seed that had detectable levels of Fusarium graminearum. That's changed since 2020. It's been seed cleaning plants that have really been, the ones that are regulating allowable amounts, because they'll say we won't clean anything with detectable amounts. So they'll say everything has to test 0%. Some seed cleaning plants say 2%. Some have even higher limits. So when you're looking at your seed testing results, and you need to know whether or not you can clean the seed, you can use those results to determine that. We do have the Alberta Fusarium Management Plan. It's kind of archived now, but you can still find it. This is kind of the recommendations that they have there. It does use kind of a risk model for recommendations. Again, it has to do whether or not you've seen Fusarium graminearum historically. And whether or not you've seen downgrading, kind of. So that can help with making a decision. As far as my recommendations go, when growers call in, or a client, whether or not to use the seed, my recommendations are, it's very clear if they've seen Fusarium graminearum in the past, or not. If they've never seen Fusarium graminearum before, I don't recommend using seed with any detectable levels, because you don't want to introduce it into a new area. You want to avoid that from becoming a problem before it is. And it really comes down to risk, and how much risk you're willing to take. Basically, the higher percentage of infection, the higher the risk it will impact ultimately the yield of the crop, but also future years, because plants grown from infected seed can serve as an inoculum for years to come. So it's really difficult to quantify, how much damage planting infected seed can do, because it's not just next year's crop that it's impacting. Numbers wise, I think it's really important to get that fungal screen, add up the Fusarium graminearum, other Fusarium species, and Cochleobolus, because these are all causing those same sorts of issues, seedling blights, root rots. And so when you have those added up, you can kind of get a better idea of what kind of impact they'll have. If you're looking at 30 to 50% in your sample or higher, this is very high risk. That's 1 in 2 or 1 in 3 seeds that you've got infected. Between 20 and 30%, this is high risk. This is 1 in 4 seeds kind of range that are infected, right? So that's quite a lot that's going into the ground that has an infection. 10 to 20, medium risk, 1 in 5 to 1 in 10 seeds that have an infection. Less than 10%, I'd say low risk. This is less than 1 in 10 seeds that has an infection, but still 1 in 10 can be quite a bit. So, it's up to the individual and how much risk they're willing to take. If you've got over 10%, you definitely want to use a seed treatment registered for the pathogens present. This depends totally on individual growers' situation, though, as well, because, like I said, Fusarium graminearum, it is more aggressive, right? So you might, if you have like 10% Fusarium graminearum, that's different than 10% Fusarium or 10% Fusarium species. So you really have to look at the individual results and make a decision based on that. So what do you do if you have disease present? You want to follow best management practices. And this all comes down to the disease triangle and trying to manipulate it so that you don't have high risk of disease. So again, 3 factors. First, we'll talk about the host. So you can use resistant varieties. So depending on what diseases are a problem for you, you can use crop types with more resistance, right? We know that generally oats are less resistant, or are more resistant to Fusarium graminearum, rather. So if you're having a Fusarium problem, oats might be a better option. Planting seed with high germination and vigor will ensure a more uniform establishment throughout the field, and that's really important. And increasing that seeding rate decreases the tillering. And again, uniform field, this allows for better disease scouting, and really importantly, reduces that window for infection at anthesis when the heads are flowering, right? and it decreases that need for multiple fungicide applications to control disease. The next corner that we'll look at is the pathogen of the disease triangle. Important to test seed, of course. Look at multiple options. Use seed with the lowest levels of pathogens present. Don't plant seed infected with Fusarium graminearum in an area that is pathogen free. You don't want to introduce it. Following crop rotations reduces the proximity to stubble-borne inoculum, allows that decomposition of the stubble, and that disease will die out over time. Allowing 2 years out of cereal and corn is really important, specifically for Fusarium graminearum. Considering field location as well, those sexual spores that are produced in the parathecia, those are they are airborne, so they can travel small distances, right? So if you're planting a field downwind from a field that was infected last year, your risk of seeing disease in that field certainly goes up. And then the last corner is the environment. So you can make the environment less favorable for disease by applying a seed treatment for the pathogens that are present that you see on that fungal screen. You can spray a foliar fungicide, at the proper timing at anthesis with Fusarium head blight. You can monitor the environment using that Fusarium risk map that I showed you earlier, and that can give you an idea whether or not you want to apply a fungicide as well to manage that risk. The Alberta Wheat Commission has a website, managefhb.ca. It's really quite useful in looking at the different management tools. They split it up by the different parts of the when you're doing the planning up at the top there, in-season management, and harvest management, and storage and handling management as well. And there's a few other points there that I didn't really go over that can be useful if you want to do everything you can to manage Fusarium head blight. So that's the end of my presentation today. Are there any questions?
Thank you so much, Trevor. I hope you all enjoyed this amazing presentation. If there's any questions, feel free to type in the comment section or the chat or the Q&A.
Sure.
I guess we should wait for a few minutes because sometimes it takes longer, and plus we've been having a really bad internet connection since morning.
Oh no, okay.
Yeah, that's why it took us longer. Like I was trying to get that fixed.
Right.
Yeah. Are there any questions?
Well, if you do have any questions, you can reach me. I'll just share this again really quickly, I guess. You can reach me at this phone number, 780-955-3435. Just ask to talk to me, and they'll direct you right to my phone. Or you can email me at trevor@2020seedlabs, and I'll be happy to answer any questions. Oh, okay, sure.
Yeah. If infection is largely windborne, why has the spread not been northward and westward?
Oh, like from the kind of prevailing winds, like from the north down to the east, or down to the south and towards the east. The reason is where the disease originated, really. So Saskatchewan, they didn't have zero tolerance like we had in Alberta, right? So and you saw previous years, Fusarium graminearum was much more common in Manitoba and Saskatchewan than it was in Alberta, because we had that Fusarium management plan, which really did work for quite a few years to help control the spread, because nobody was planting infected seed, right? So not all wind goes comes from the northwest, so you will get spread field to field eventually coming from other directions, and Saskatchewan was a source of infection, and that's why we're seeing it spread from that eastern border. Um, from the south, I think, there's kind of a band there that's differentiated between southern Alberta and up towards the Edmonton area, where there is a lot more pasture, right? And that does help to split up where the infection can come from. It doesn't serve as a host for Fusarium graminearum, of course. So that kind of protects from the south, where we saw Fusarium graminearum show up first, possibly because they had a lot more corn acres down there. They have more durum, they have more susceptible crops. There's irrigation down there as well, so even though we do see the environment being an important part of determining whether or not you've got a lot of positives. If you're irrigating, that will might not have as much of an impact in those areas, but we still are seeing quite a big effect from the rainfall in those areas.
Perfect. There's another question by Krish. Thank
Okay.
you for the presentation. Are these FG levels started in the seed bags too?
Started in the what, sorry?
Hello? Oh, I'm so sorry. Uh, are these FG levels stated in the seed bags too?
Stated in the seed bags.
Yeah.
Like if you buy some seed, whether or not it's reported on there, um, it not necessarily. Fusarium testing isn't a requirement for buying pedigreed seed, right? So all having pedigreed seed says is that it meets those certain requirements. The one thing that does have to be done is a smut test, that true loose smut test, right? Because that is on the grading tables. So there's no, as long as the seed meets those requirements for germination and all the other requirements, then you're not then it could still have fusarium, certainly, or other diseases that are present that is not reported because of that. So if you are buying pedigreed seed, it is a good idea to ask for those disease results to see what the disease profile that's present is. And if it needs a seed treatment or other decisions that you can make.
So there is another question. To what extent does direct transfer of infected non-grain seed material, such as infected stubble, on equipment result in the spread?
Oh, I'm not too sure if that's like if it's probably not a huge amount. Not like soil-borne pathogens would be spread like clubroot is spread by soil moving on equipment in a major fashion. But there is a possibility, if you do have like a lot of infected stubble present on equipment, there is a possibility, and it does fall off in another field. There's probably a low risk of that, but it wouldn't be a major source of spreading at longer distances, I wouldn't think. An infected field, there's a lot more stubble there, right? There's a lot of infection, a lot of potential inoculum compared to the amount that would be moved on equipment, I would think. But it is a possibility if like there's some sort of way that something's spread that you're not quite sure. In theory, it could happen, I suppose, but I wouldn't consider it a major risk.
Perfect. Are there any other questions? I guess let's wait for another minute or two
Okay.
and There's no questions then. Yeah, you were stucking sometimes, like when you're trying to answer questions. So I don't know if it was for everyone else too.
Oh, hopefully not.
Mm. Yeah,
But yeah, if there's anything that wasn't clear that during the presentation or in the questions, yeah, please follow up with me, and I'll try to clarify everything as best as I can.
I guess there's no more questions then.
Okay.
Okay, yeah. Thank you so much, Trevor, for addressing these questions and delivering an excellent presentation. It was truly a pleasure to have you with us today. And to all the attendees, we appreciate your participation in today's webinar. We hope you found the presentation informative and enjoyable. This marks the end of our session. Thank you once again for joining us. If you have any further questions or feedback, feel free to reach out to us. Have a wonderful day. And I'm so sorry again for really bad internet. Yeah,
All right. Thank you for having me, Kabir. It
yeah.
was good to be
Thank you
here
so much.
today.
Yeah,
All right.
thank you so much.